Hello, I'm Fergus Shanahan, speaking to you virtually because of COVID restrictions, speaking to you from APC Microbiome Ireland, a research centre funded by Science Foundation Ireland, dedicated to the microbiome in health and disease. Now, I've been asked to speak on the topic of, can the microbiome improve our lives in the future? And normally I dislike speaking about the future because I can't project to the future, but in actual fact, in the case of the microbiome, I think there are already success stories that I'd like to discuss that do suggest a very bright future for microbiome improving our lives, human lives in the future for all of us. There are some disclosures to present to you. None of these have any constraint in what I'm about to speak on, and none of these influence the content of what I'm about to speak on. Now, almost a decade ago, the then president in the United States said that for every dollar we invested to map the human genome, it returned 140-fold to the economy. If this is true, then it would suggest that the much larger microbiome, that is to yield far more, not just economically, but in terms of health benefit to humans. Because the microbiome is vastly greater than the human genome. And more importantly, it is tractable. That is, it is susceptible to manipulation and changes, and we have some control over it for good. Now, it's almost impossible to read a medical journal today without seeing some mention of the microbiome in relation to, other than perhaps trauma, that seems to be unrelated to the microbiome. And so my colleagues and I felt obliged to write an article recently criticizing some of the strategies and techniques being used to imply causality, when in actual fact only correlation was being shown. So there is some issue about the technicality and the quality of some of the studies, but nonetheless, there have been quite dramatic success stories that I'd like to remind you of, from which there. And these are all success stories with the human microbiome, with human disease. I won't be discussing in large part studies in experimental rodents. I'm mainly speaking about human advances. And of course, the first one must be the discovery of Helicobacter and its relationship to what seemed at one time to be highly heterogeneous diseases. But nonetheless, a single solution was found, namely a specific microbial species identified, which if removed, actually changed the whole picture and made diseases like peptic ulcer disease almost a historic thing, at least in young people in the Western world. And we used to talk about gastric cancers being highly heterogeneous, but yet the removal of Helicobacter from the picture has actually contributed to a tremendous decline in gastric cancer. But there were other lessons from this discovery that portend a good future for us. The first and most important lesson is that there are some diseases which will never be solved by research focused exclusively on the host, unless one actually starts to look at the environment, particularly the immediate microenvironment. The discovery also showed that while we speak of non-communicable diseases in terms of the non-transmissibility of these diseases, Helicobacter shows that actually risk of these diseases can be transmitted, particularly in relation to the microbiome. The discovery also showed the importance of context. It's not appropriate to speak of good and bad bacteria. In fact, Helicobacter can be a friend as much as a foe. Look at this slide where we portray on the bottom part of the slide the risks and costs associated with Helicobacter depending on one's age. Not all of us will get gastric cancer or lymphomas or ulcers. But yes, it is indeed a major risk factor for gastric cancer. In fact, it's a class 1 carcinogen. But on the other part of the slide, you see that Helicobacter has almost certainly been a protective factor at some stage during the development and evolution of humans. And it isn't just about the cancer of the stomach and the body of the stomach. Helicobacter is now almost certainly a protective factor against reflux-associated cancer at the upper end of the stomach and the lower end of the esophagus. So there's this yin-yang about individual organisms. It's no longer acceptable to just speak in terms of good and bad with an organism. One has to have a broader perspective. The second success story that I would identify is the whole area of Clostridium difficile and fecal microbial transplantation. Now, that's not a new discovery. That has been around actually not just for centuries, but millennia prior to even the identification of Clostridium difficile. This microbial transplantation was actually being practiced. But it does open up a whole new area. It's the ultimate microbial experiment, if you will. And I've just tried to plot here the whole idea of manipulating the microbiome or using, if you will, bugs as drugs. And the precision of action increases as one goes from the very crude whole microbial transplant all the way up to single strains and then products from those single strains, including phage and including antimicrobial bacteriocins and perhaps other metabolites. I showed a slide to make the point that the future will not be about fecal microbial transplantation. It will almost certainly be about using selective consortia or single strains that we've identified and verified that are free of pathogenicity islands and free of the potential to transmit antimicrobial resistance. So the future will be a more designer stool, if you will, designer consortia of organisms for not just Clostridium difficile, but many other conditions, including patients undergoing cancer chemotherapy that I will allude to just shortly. For those who are loose in their use of fecal microbial transplantation, one has to make a point about the lessons we've already learned. We already know from experimental animals that simply transferring or transplanting microbes from one animal to another can confer, at least in experimental animals, different phenotypes, including the potential for the generation of inflammatory phenotypes, metabolic changes, and even behavioral changes. And as I'll show in a moment, there are risk factors within the microbiome that may increase or decrease one's risk of colon cancer. So this has to be considered in addition to safety risks such as transmission of infectious diseases, but the potential to transfer phenotypes must be considered if one is going to loosely jump to fecal microbial transplantation. And hence my suggestion that we will almost certainly have to refine this rather crude therapeutic strategy. But the prospects are excellent and we already have proof of principle. My third success story for human therapeutics is the whole area of nutritional science. The microbiome is redefining what we call dietetic and nutritional science. In fact, it's leading to a revolution in these areas, and it's underpinning high quality nutritional science. I'll give one or two examples of this. My first realization of this is summarized in this slide when we published in Nature some years ago, a study of elderly individuals going into long stay residential care. And what happens there frequently is that their dietary diversity collapses. As people age, their dentition and other physiological changes pushes them more towards a more monotonous diet. And this is often formalized when they go into a long stay institution where they get adequate calories and nutrients, but lack of diversity. They go for more milk-based diets. When we followed this, we found that leads to a loss of microbial diversity. So dietary loss of diversity happens first, then one gets loss of microbial diversity, and that has been associated with objective measures of inflammation, so-called inflammaging, which accelerates the aging process and actually was associated with objective measures of frailty in our study. There has since been intervention trials to reverse this, whereby European subjects have actually been trialed with a Mediterranean diet, which actually reversed these microbial changes and halted the progression of frailty in long-term studies. So dietary diversity is not just staple, it's spice of life, and it is no longer acceptable to simply do a nutritional assessment of an elderly person for traditional measures of caloric intake and nutrient intake. One has to factor in diversity as well as the traditional content measures. And this is a challenge for the food industry. I predict we will have new functional foods, new foods designed specifically to cater for the physiological changes and impediments and constraints that are imposed by aging to try and confer this dietary diversity for welfare. We're trying to improve healthy aging, not necessarily expand the duration of life, but rather expand the period of time where one has a healthy age spectrum. And this particular slide summarizes a recent review where we've looked at various aspects of nutrition, which are now underpinned and explained by the microbiome. And in essence, the microbiome is the transducer of nutrient signals, and nutrition will have to be considered in that context in the future. And it will vastly improve the prospects for a predictive microbiome and a personalized microbiome based on personalized diet. The fourth area, which I think is a success story and which will improve our lives if we're unfortunate enough to develop cancer, which has a high risk in aging populations. And I believe the microbiome actually comes to our rescue here also in relation to being a source of biomarkers for risk of cancer, but also as a source of microbial factors which can modify our clinical prognosis and response to cancer chemotherapy. Now, some of this has been obvious to clinicians for a long time. I've already mentioned Helicobacter as a single species that can serve as a risk factor for gastric cancer, and which, if eradicated, removes that component of risk. But what about the small bowel versus the colon? It's been evident to clinicians for a long time that small bowel cancers are extremely rare in contrast to large bowel cancers. So this would suggest that the microbiome is causative, perhaps not a single strain linked with the cancer, but rather combinations or consortia of strains of microbes that might increase the risk of cancer or perhaps in some individuals protect against large bowel cancer. The slide tries to also show that the host has a say in this matter. The way in which the small bowel handles microbes differs from that in the large bowel. The small bowel is adapted to nutrient intake and actually allows ingress of metabolites, and it protects itself by producing antimicrobial regulatory peptides from the epithelium, whereas in contrast, the colon does it structurally. It has a different structure to its mucin, whereby the inner layer of mucin protects against ingress of bacteria. But unfortunately, in a westernized diet, in an industrialized microbiome that we have in the Western world, we actually have fostered microbes that tend to digest the mucin layer, and which would, in a non-industrialized society, would actually be metabolizing fiber, but in our society is actually now resorting to metabolizing our mucin layer and increasing the exposure of the epithelium to commensal bacteria, some of which may be risk factors for colon cancer. There are experimental models of this in surgical arenas. We've already known for a long time that when ileoanal pouch anastomoses are constructed, where one has a colonized small bowel, if you will, in some patients who undergo this for either polyposis coli or ulcerative colitis, that the pouch rarely, if ever, develops a cancer, even though it is colonized. So it isn't all about the microbes, it's the host response that's important. And in my experience, the only time I've seen a cancer in these individuals is when there's some cuff of colonic mucosa inadvertently retained by the surgeon. So we set out, like others, to look at the microbiome as a source of potential risk factors for colon cancer. And we extended the observations of others by taking feces, but also taking mucosal adherent microbes, not just on colon cancers, but associated with colon cancers and distant to colon cancers in the same individual. And this is the design of an ongoing study where one can look at what organisms are there, what they're doing metabolically, and what potential interaction they might have with the host. And this has been in a series of 2 papers published in Gut recently. And the summary of the findings is as follows. The microbiome is indeed, as others have found, linked with colon cancer. the changes in the microbiome associated with colon cancer are not restricted to the cancer. We find them throughout the colon. So it's a field change in microbes, even distant, proximal or distal to the actual tumor lesion. The important part of this slide is that we found also changes associated with individuals with polyps. Now, that's important because most cancers are derived from polyps, not all, but most cancers are derived from polyps, although most polyps do not necessarily cause cancer. But if we can identify a microbiome change associated with a pre-malignant lesion, then we have for the first time microbial biomarkers of risk. And most of the in fact, all of the biomarkers we have for colon cancer at present are actually markers of early disease, established disease, such as the appearance of blood. But now this, for the first time, is a biomarker of risk of disease. Curiously, we found that some of the bacteria associated with colon cancer are actually normally found in the oral cavity. And when we combine the oral microbiome with the fecal microbiome, we actually achieved the sensitivity and specificity for the diagnosis of colon cancer, actually that rivals some of the non-invasive immunotests that are currently being used in national screening programs. But our purpose for doing this is not for diagnosis. Rather, it is to identify people at increased risk so that they can be selected specifically for advanced screening by colonoscopy. One can take this a step further, as this bright postdoctoral student working with us has done, where she's taken some of these fecal microbial samples, put them into germ-free animals, notobiotic animals, and effectively humanized those animals, and has been able to show that some of the microbiomes that are associated with high risk of colon cancer actually lead to suppression of the host immune response, and vice versa with the low-risk microbiota. So we're getting to a stage where we can start to understand how it engages with the host immune system and interfere with this at an early stage by actually doing something to individuals at high risk, such as a dietary change or a microbial transplantation. So that's the future for people at high risk with colon cancer. Now, what about other cancers? It's a decade since this paper appeared in Science, about 10 years ago, showing that irinotecan, one of the chemotherapeutic agents used for many cancers, is actually metabolized in the gut, and the microbiome actually reactivates an inactive drug when it's been excreted, and that explains some of the toxicity and side effects with this drug. But it has been possible to interfere with the microbiome to actually restore the activity of the drug without having the toxicity. Again, that's the future where microbiome is leading us. Along the way, this paper appeared that I think people, it was underappreciated and many people missed it. This was showing in a cancer center in patients undergoing allogeneic stem cell transplantation for various cancers such as lymphomas and leukemias, that when it was looked at their overall survival or their transplant-related deaths, these are hard data now with Kaplan survival curves, showing that for one measure of microbiome health, namely just diversity, you could see a complete separation of patients in terms of their survival, just if one used one particular marker, microbiome diversity. Nowadays, we'd use more sophisticated tests of microbial composition, but for diversity alone, look at the extreme separation of these individuals. This demands that the microbiome be looked at in terms of a modifier of our cancer therapy for our patients. And there have been many studies now showing that for some of the more modern cancer immunotherapies, such as the checkpoint inhibitors, that one can explain in experimental animal models how the microbiome is enhancing or suppressing the responsiveness of an individual to therapy with a checkpoint inhibitor. Not only that, in human studies now, this is a paper from Nature Medicine, and not too long ago, a couple of years ago, linking individual taxa or particular species where they're known, individual microbial species and strains linked with responses or non-responses to immunotherapy. That's where the science is actually taking us now, where we could have baseline assessments of whether someone is likely to respond or not respond to checkpoint inhibitors. But the story gets better, because for people who might be cancer chemotherapy resistant, the story improves for them also. here are two papers in Science at the end of the past year, within the last few months, showing that fecal microbial transplantation in the unfortunate individuals with melanoma who are undergoing immunotherapy, for those who are resistant to treatment, it was possible to restore their sensitivity and their responsiveness to the treatment by doing a fecal transplant. Now, as I say, these data will almost certainly further improve as we start to restore the favorable bacteria and leave aside the non-favorable microbes. I'll close on something else. I'll close on the potential to develop drugs from bacteria and other microbes in the human gut. And we and others have identified that numerous microbes already present in the human intestine, largely in the colon, are a source of antimicrobials such as bacteriocins and phage, which can be used as antimicrobial factors. They are a source of anti-inflammatory metabolites and components of exopolysaccharides and internal metabolites and cell wall fragments that are anti-inflammatory, that can actually be the next drugs, and associated immunomodulatory factors. But I'd like to show you some data, recent data, that has all gone all the way to human translation. And this is the production of a growth factor from Bifidobacteria that are shown on the electron microscope on the left here, where we've been able to show that Bifidobacteria, which are the first colonizers in a newborn baby and therefore likely to be safe, this is showing that when Bifidobacteria appear in the human intestine, they cause the epithelial proliferation, because the human gut sends a signal to these Bifidobacteria to express these pili, or hair-like particles on the surface, on their surface, and they in turn express a molecule called TAD-E. The detail is not that important, but the essential point is that TAD-E, once it's upregulated in vivo, it's never seen in vitro, so you'd only find this in human in vivo experimentation. Once the TAD-E is expressed, it induces proliferation of the epithelium. Why is that important? Because it can accelerate closure of the microbial barrier in preterm babies, or it might be used as a new drug. One can actually visualize some of these molecules on the surface of these pili that the Bifidobacteria expresses. And here's a study inducing ulcers in mice, experimental mice, where one induces wounds by biopsies. And you can see the Bifidobacteria, if they express the TAD-E, they cause healing because of epithelial proliferation. But if we delete the TAD-E protein from the surface, one gets no healing. Now, I'm not here just to show you murine colonoscopy. We went on to look in a series of humans where we induced ulceration by aspirin in the small intestine, and showed that co-administration of the organism expressing the TAD-E, one got healing of this very, very common ulceration that occurs in humans when they're taking aspirin for various reasons. This doesn't just apply to aspirin, of course. This is relevant to the mucositis associated with cancer chemotherapy as well, and numerous other potential indications. So in closing, although I was reticent about accepting this task of speaking on how the microbiome could improve our lives in the future, I would respond by saying, on close consideration, the microbiome is already improving our lives, and there are multiple success stories that I've outlined here that will show already that the microbiome has improved the lives and welfare of humans, but promises much more if we can learn the lessons from what we've already seen with these success stories. Thank you all for your attention.